We've seen how cells divide and learned the phases of the cell cycle, but we haven't talked about when or under what conditions cells divide. That's what we're talking about here, cell cycle control, and a problem that arises when cell cycle control breaks down, namely cancer. Let's start by describing constraints on cell division. Basically, cells only divide in certain situations. The normal state of most cells in adult human is not to divide. Take, for example, the top of the figure, which depicts a normal situation. These cells are not dividing, and there are a few features that keep them in their living but not dividing state. Note first that they are attached to a surface. In the body, this usually means another kind of cell or extracellular matrix. For example, the upper layers of your skin grow on top of a lower layer of connective tissue. In the laboratory, this is replaced by a plastic petri dish, and in that case, they only grow to be one cell layer thick. So cells are anchorage dependent. They need the proper surface to grow on. Now let's take a look at what happens when this layer of cells gets damaged. For example, if you cut yourself. Injury leads to a loss of cells and an opening in the skin. Those cells that are displaced die off because, once again, they are anchorage dependent. If they're not in contact with the appropriate surface, they die off. But the injury stimulates other cells to grow. So the cells surrounding the injury start dividing. They will continue to divide until the missing cells are replaced, and then they stop dividing. That's because when cells start to touch one another, cell division stops. This is called contact inhibition, or density-dependent inhibition. It's the reason that new skin doesn't just keep growing when an injury heals. New tissue grows just enough and then stops, or contact inhibition. Cell division is also under the control of growth factors, protein signals that, as the name implies, tell cells to start dividing or growing. More than 50 growth factors have been identified. So when the injury happens up here, growth factors are secreted, and these cells tell these cells to start dividing. So normal cells are growth factor dependent. That is, normal cells don't divide unless they're told to do so. What I've described so far is all normal biology. But in cancer, these constraints on cell division break down. In other words, cancer cells are anchorage independent. They're not contact inhibited, and they're growth factor independent. Before we talk more about cancer, let's talk about how the cell cycle is normally controlled. The cell cycle proceeds through a series of checkpoints, checkpoint, 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 which stop cell cycle progression unless a set of conditions have been met. Let me describe this concept of checkpoints by saying it is the opposite of dominoes, another possible mode of control. Imagine you take the time to set up a long line of dominoes. When it's time to knock them over, you just knock down the first and the rest fall in turn. If cell cycle control followed the same logic as dominoes, the cell cycle would start and go all the way through to the end when the cell was replicated. This clearly is not how cell cycle control works. Rather, cell cycle proceeds through a series of checkpoints. At each checkpoint, cell cycle progression will stop until certain conditions have been met. What those conditions are vary from one checkpoint to another. You can think of these checkpoints as command and control of cell cycle. There are 3 major checkpoints, and we'll look at each one separately. But for now, let's just see when in the cell cycle they act. And it's easy because their name tells you when they act. There's the G1 checkpoint, and it's active in G1 here. Note that the G1 checkpoint kicks in before DNA replication, so the cells doesn't waste time or energy replicating DNA if it doesn't have to. Some cells do not divide because they're mature and the body just needs them to stay alive and do their job. Nerve cells, for example, don't normally divide. Cells like this that have left the cell cycle are said to be in G0. The G2 checkpoint lies between G2 and mitosis. Your textbook calls it the G2 checkpoint, but most other sources call it the G2M checkpoint, precisely because it lies between G2 and mitosis. In other words, it stops the cell from entering mitosis. The M checkpoint stands for metaphase. Recall that in metaphase, chromosomes align along the equator of the cell. This turns out to be very important because if chromosomes don't align properly, they don't distribute to the new cells properly, and that's a disaster. So the M checkpoint stops the cell cycle unless chromosomes align properly. Now let's talk about cancer. It's the second biggest, or maybe even the biggest, killer in the US, and therefore we should take the time to study and understand it. But we're going to talk about only those aspects of cancer that can be explained by defects in the cell cycle. That's consistent with the idea that cancer is the uncontrolled growth of cells. But that's kind of an old-fashioned and simple view of cancer. We now know that there's a lot more to cancer than uncontrolled growth. Cancer cells also defy death. That is, they don't die off under conditions in which normal cells would die off. They can change their identity and travel to other parts of the body. These are all factors that make cancer a deadly disease. For this class, however, we will just consider the growth aspects of cancer because they tie in very closely to our discussion of cell cycle control. A moment ago, I said that normal cells are growth factor dependent. Cells don't divide unless they receive a signal to do so. The G1 checkpoint is responsible for this constraint on cell growth. That is, cells are arrested in G1 unless they receive a growth factor signal. A growth factor signal is a protein secreted by another cell that binds to the surface of the cell receiving the signal. The signal goes to the nucleus, where cell division is initiated, and eventually the cell divides into two cells. So growth factors overcome the G1 checkpoint. Remember that the G1 checkpoint comes before S phase, so cells don't waste energy replicating their DNA without a growth factor signal. Remember also 2 slides ago we talked about anchorage dependence, contact inhibition, and growth factor dependence. These are all events controlled by the sort of signaling you see here, and so are all mediated by the G1 checkpoint. As I've already said, cancer cells are anchorage independent, not contact inhibited, and growth factor independent because of a failure of the G1 checkpoint. We've already said the G2 or G2M checkpoint, I will say G2M from now on, stops cell cycle progression before mitosis. Let's think about what's going on before mitosis, and then what the G2M checkpoint does will make more sense. Before G2 is S phase, when DNA gets replicated. DNA replication generally includes some mistakes or mutations. The product of DNA replication is 2 molecules of the original DNA. But the replication process is not perfect and errors are made along the way. That's what these red stars are meant to imply. Errors of DNA replication or mutation. We've already learned that during G2 the cell is repairing any damage that happens to its DNA, including mutations due to replication. There are other sources of damage to DNA, like radiation or chemical toxins, and the cell generally does all it can to repair that damage. But with G2 coming between S phase and mitosis, it's ideally situated to fix errors due to replication. It should make sense then that if DNA damage is too great and can't be repaired, the G2M checkpoint kicks in and prevents the cell from entering mitosis. So the G2M checkpoint halts cell cycle progression in the face of irreparable DNA damage. The G2M checkpoint has a second role. It stops cell cycle if the cell is too small. Basically, the cell has to grow to a certain size before cell division can happen. Otherwise, the resulting daughter cells will be too small, lack enough organelles, to survive. Putting it all together, the cells have to have repaired their DNA and reach a certain size for cell cycle to proceed. And this control is mediated by the G2M checkpoint. Cancer, as we've already said, is marked by a failure of cell cycle checkpoints. Failure of the G2M checkpoint has certain predictable consequences: an accumulation of mutations and other DNA damage, like broken chromosomes. And that's exactly what we see in cancer cells. A hallmark of the cancer cell is damaged DNA with lots of mutations. This is a video that you've seen before, but I'm showing it again because it shows pretty clearly the M checkpoint in action. Remember that M in this case stands for metaphase. I know it's a little confusing because in other cases M stands for mitosis. It's just a confusion we'll have to live with. Remember also that metaphase is when chromosomes align along the cell's equator. Let's roll the video and we'll see exactly when this happens. Okay, so here's the outline of the cell. This whole thing is the cell, and you can see the nucleus inside the cell. And all of those are chromosomes within the cell. They're visible and condensed, so we're in prophase. All those chromosomes are attached to the mitotic spindle, and the mitotic spindle is trying to get them into alignment. So that's the M checkpoint in action. All these chromosomes have to come into alignment before anything else will happen. And you'll see this one right here is the last to get into a line. Okay, so that's finally getting into a line, and when everything is perfectly aligned, everything then happens quickly. Okay, anaphase, which you just saw, followed by telophase and cytokinesis. The critical importance of the M checkpoint is that if chromosomes don't align properly, they don't segregate properly, leading to daughter cells with too few or too many chromosomes, and cells can't generally survive that situation. As with the other two checkpoints, we have to talk about what happens in cancer cells when this checkpoint fails. As you'd expect, failure of the M checkpoint leads to an abnormal number of chromosomes, and you see this a lot in cancer. The number of chromosomes can be way off in cancer cells. I know I just said that cells can't survive with too many or too few chromosomes, but cancer cells are different. They've been selected to survive these conditions. Failure of both the G2M and M checkpoints combined lead to genome instability, another hallmark of cancer. Okay, now that I've set the stage for studying cancer, let's talk about what actually happens when a normal cell becomes a cancer cell. Just to be clear, it's a long process and it happens in many steps. It also involves a lot of terms that will be new to you, so heads up. The first step is transformation. That's when a normal cell first takes on the properties of a cancer cell, especially uncontrolled growth. Note that it is mutation, or an accumulation of many mutations, that leads to transformation. A transformed cell is one that reproduces or proliferates uncontrollably. That's called hyperplasia. It simply means cells are growing more than usual. This leads to a tumor. A tumor is a mass of transformed cells. Remember that transformed cells are not contact inhibited as normal cells would be, and are anchorage independent. That's why they can form tumors. Normal cells don't form tumors. Tumors can be benign or malignant. Benign tumors tend to stay put while cancerous tumors invade other parts of the body, a process called metastasis. A benign tumor is one that doesn't spread. That doesn't mean it's not dangerous. Imagine a benign tumor in the brain or other vital organ. That's a dangerous situation because it could impair the normal function of that organ. A malignant or metastatic tumor is one that is capable of invading other parts of the body. At first, the invasion is local, spreading to neighboring tissues, but the tumor may grow enough that some cells slough off and enter the bloodstream. Of those that enter the bloodstream, some will take up residence in another part of the body and begin to grow there in a secondary tumor. Again, this process is called metastasis, and that's generally when we call the disease cancer as opposed to a benign tumor. Most cancer death is caused by secondary tumors. Since I've talked so much about cancer, I should say something about cancer treatment. First though, let me clarify something. I've been talking about cancer as if it's one disease, but it's not that simple. There are more than 100 different kinds of cancer. Lung cancer, for example, is very different than ovarian cancer, and there are at least 3 different kinds of lung cancer. Correspondingly, cancer treatment varies depending on the type of cancer it is, where it is in the body, and how far it's progressed. You may be aware that a revolution is underway with regard to cancer treatment. Immunotherapy is the idea of using the body's own immune system to fight cancer. Cancer cells are very good at evading the immune system. Immunotherapy attempts to block that ability in cancer cells. Drugs that take this approach are very specific for a particular cancer, as opposed to older drugs that are not as specific and have lots of side effects. Here's an example. The round yellow cell in the picture is a cell of the body's own immune system. It has been engineered to recognize tumor cells, like the one in green. In this case, the immune cell attacked the tumor, which then underwent cell death. Cell death, or programmed cell death, is when a cell dies off, kills itself really, in a very orderly way. The remnants of the cell, these green blebs, can now be removed by other immune cells. I'm not going to say any more about cancer immunotherapy or any of the other newer specific cancer treatments. I will concentrate on the more conventional forms of cancer therapy. Keep in mind, though, that these newer treatments are typically second and third lines of defense after more conventional therapies have failed. While they vary a lot from cancer to cancer, there are some general themes for treatment of cancer. First of all, surgery and chemotherapy remain the backbone of cancer treatment, and how cancer is treated depends on how advanced it is. In the early stages, localized tumors are removed surgically or treated with localized beams of radiation. The idea is to cut out or kill the tumor before it spreads. The surgery part has been true for hundreds of years. Highly localized radiation therapy is relatively recent. Attacking the primary tumor with surgery or radiation is often accompanied by chemotherapy. Conventional chemotherapy is somewhat nonspecific in that it kills not just cancer cells, but many other rapidly dividing cells. One way they do this is by halting the cell cycle or inhibiting the mitotic spindle. Because it's nonspecific, side effects occur in other rapidly dividing cells, like intestinal cells. As you may know, one of the big side effects of chemotherapy is nausea and vomiting, which results from killing these intestinal cells. To recap, in this video we learned that cell division is under very tight control and that defects in this control contribute to cancer. In the next video, we'll learn about a specialized form of cell division, meiosis, and how it leads to genetic diversity.